My name is Graeme Milligan. I'm the Gardner Professor of Biochemistry at the University of Glasgow, and I'm also the co-director of the Centre for Translational Pharmacology.
Hi, I'm Natasja Barki. I'm a postdoc in the Milligan lab, and I'm working mainly on the gut function and how short-chain fatty acids can affect both enteric and peripheral changes.
In terms of the broad efforts of my laboratory, I'm really interested in the idea that many molecules we think of as part of our food can actually act in a hormone-like manner to really improve our health. And this then really feeds into issues around pre and probiotics and what the microbiota in our gut do for us as they've co-evolved with us over millions of years. And Natasja is focusing on particular molecules that are generated by our gut microbiota in response to the fiber in our diet. So short-chain fatty acids are things like acetic acid, you know, the vinegar that you put on your fish and chips. And we don't really think of that as a hormone, but actually in our lower gut, the microbiome generate very large amounts of acetate and a related molecule called propionate from metabolism of our foodstuffs. Some of that gets used locally in the gut, but also after it's taken up into the bloodstream, it regulates a number of our key organs, including the pancreas that's involved in insulin secretion, the adipocytes that store fat and of course are important players in generating a whole series of hormones themselves, and indeed the white blood cells that provide the front line of our immune system. So the paper was really all about trying to understand which specific receptors in those tissues generate the effects that are produced by these short-chain fatty acids as and when the gut metabolizes fiber into these molecules. And we tried to use a clever methodology by generating mice in which we'd introduced a modified form of one of these receptors that's called a DREAD, which stands for designer receptor exclusively activated by designer drugs, so that we can specifically focus on functions of that one receptor type. And Natasja was at the heart of doing many of the experiments behind this set of studies.
One of the main findings within the paper itself, as Graeme already mentioned, we looked at how activating these receptors can affect changes within the gut, like the release of hormones. But one of the main findings was that it doesn't just affect changes within the gut, but can also affect changes in your central nervous system. So one of the findings was that if you activate these short-chain fatty acid receptors in the gut, neurons or cells in the spinal cord will also be activated. And the reason we were able to achieve this outcome was using our transgenic mouse line, as Graeme already described, our DREAD mice.
And one of the intriguing things about the idea of this link between what we eat and how we behave is there's been a great fascination now for a number of years in the idea that the gut and the brain communicate bidirectionally. There have even been some suggestions that what we eat can regulate central nervous system disorders ranging from depression to Parkinsonism. And although that can seem a little bit like science fiction and is often quite observational, what Natasja's been trying to do is bring a mechanistic and biochemical understanding to those observations that people have made at a medical level. Indeed, and that's one of the things we're trying to test directly, in fact, Ali. The idea that we could, rather than taking traditional antidepressants, for example, give treatments that regulate the receptors that respond to the hormones that our microbiota make, and whether they might actually alleviate a number of central nervous system disorders. Depression might be an obvious one, but clearly some of the longer term neurodegenerative disorders, and I've already highlighted Parkinsonism, but there are also thoughts around autism and various other disorders. Yes, we genuinely believe it might be possible to target in this, perhaps what appears to be rather an indirect way, but one that might be better for us in a holistic sense. As I mentioned earlier, the concept of a DREAD is simply taking a receptor protein, modifying it so that it doesn't respond to natural ligands, but instead responds to molecules that we've identified by one means or another that don't normally have an effect in the body. So this strategy has actually been used predominantly for receptors that work largely in the central nervous system. And there are many, many studies using these DREADs. One of my colleagues, Andrew Tobin, uses this very much in his studies on neurodegeneration and the possibility of finding novel strategies to treat dementias such as Alzheimer's. So clearly there are ways and means to adopt this and adapt it to many strategies in biochemistry.
The collaboration that we had with Glasgow University was extremely important for the project, because one of our collaborators, for example, John Rodell and Dave Hughes that are on the paper, were really important in basically allowing us to use a specialized technique to study the gut-brain axis. So they were the ones that had the idea. So the electrophysiology was something we did with John Rodell, where the equipment was set out. He's an expert in electrophysiology, so his expertise was really important within the project. That group itself works a lot on diseases or function of the spinal cord. So getting that help and adding that to our project to really put something together that might be really important in terms of this bidirectional gut-brain axis was very vital for the project.
And I think just to build on that, someone like Natasja is absolutely excellent, but even she doesn't know all the advanced techniques directly from her background. So learning from others, both locally in Glasgow, through the SULSA network in different parts of Scotland, and throughout the UK and then internationally, is absolutely intrinsic to what we do. Now, from the paper that you were mentioning, Ali, you highlighted that we've collaborated with a team based in Denmark, and what they have brought to the story is they're medicinal chemists. So I've worked with the leader of that group, Trond Ulvin, for many years to develop new ligands and new tools to understand the function of not only these receptors, but a variety of other receptors that are within our portfolio. I've also had over many years very strong collaborative links with the pharmaceutical industry, and actually the identification of the ligands that Natasja used to selectively activate the DREAD receptor came from a collaboration with AstraZeneca in Gothenburg in Sweden, and they actually really put together the molecule set that we tested to find the right molecules to move forward. So collaboration, as you've heard, from literally down the corridor to elsewhere internationally has been vital to this project and to virtually everything we do at the centre. So in terms of the Mullard Award, which I was given in 2022 by the Royal Society, the spin-out companies that they're referring to are two that came here from our work at University of Glasgow. The first of them is called Caldan Therapeutics, and it's called Caldan because the Cal is Caledonia, as in Scotland, and the Dan is Denmark, as in Denmark, because as I've just said to you, my colleague Trond Ulvin is based in Denmark, and it was a company that he and I co-founded to develop drugs to treat both initially type 2 diabetes, but now a disease of the liver called non-alcoholic steatohepatitis, which is a bit of a mouthful even for me to say. And that company was founded about 7 years ago now. The second company, which is much more recently, which I co-founded with two of my colleagues in Glasgow, Andrew Tobin and a chemist, Andrew Jameson, is Celtic Pharma Therapeutics. And although we do some work around neurodegeneration and indeed chronic asthma, the main focus of that company is actually finding new drugs to treat malaria. It's a relatively new enterprise, but it's going very well and we're delighted by it. But I think in relation to the specific award and this idea of supporting the biopharmaceutical industry globally, ever since I moved to Glasgow, which is more than 35 years ago, I've developed really strong links with both multinational pharma companies, GlaxoSmithKline, AstraZeneca, many others, and with small spin-out and startup companies, particularly in the US, where I licensed technologies to them that have then allowed them to progress to be much more effective and to deliver medicines to the community. So it's been an exciting time interacting with pharma companies. And of course, they have much broader insights often into disease areas than I have individually.
Next steps after this paper are basically to extend the project. So what we're trying to do is basically look how short-chain fatty acid within the gut can also have other effects, such as effect on the immune system. So how can the body response change to a pathogen or an infection if we stimulate certain short-chain or certain receptors? What we're also trying to do is extend our research into human tissue. So we have a new collaboration that we have formed with the biorepository at the Queen Elizabeth University Hospital. And what we want to do is extend our research going from a mouse model to human tissue and see whether the response that we have in our mouse model can be translated further. I think that would be a next important step that we're really looking forward to.
And I think Natasja summed that up beautifully, Ali, because for all of our projects, of course, we want to understand the basic underpinning mechanisms behind the way that various hormones work. We utilize preclinical models, particularly transgenic mice models, to really try to then understand in a physiological context how we can tackle challenges. But of course, importantly, as Natasja has highlighted, we then need to move into human tissue before we could even think of trying to develop new medicines to treat various diseases. So we try to have a seamless link between really basic biochemistry and pharmacology to the idea that we're going to develop new medicines. And of course, my own interests are taking lots of receptors that are relatively poorly characterized and really trying to determine, either through genetic associations or just our understanding developed from years of working on them, how they might be involved in disease and how we can use them to develop new medicines to tackle those diseases. They might range from, as Natasja said, lower gut inflammation through metabolic disorders to diseases of the central nervous system. And we're trying to work on virtually all of those at the moment.